Hello, everyone. Today, or in this lecture, we're going to be discussing nucleotides and nucleic acids. I feel like this is a natural progression from what we were just talking about, that being sugar structures. And my reasoning for that is we talked about a number of different sugars. We talked about cyclic sugars, and we talked about um, aldoses and ketoses. And the first sugar that I want to go ahead and start out with is actually a cyclic sugar that looks a little bit like this. To count up the numbers, I've got my 1, 2, 3, 4, 5 carbons. Okay, so there's my 5 carbons. What I also have is carbon number 1 is a newly formed stereocenter or newly formed chiral carbon. And I'll just go ahead and put my hydroxide group going up. Then on carbons 2 and 3, I'm going to have hydroxide groups going down. Now, this sugar is basically the cyclic version of this sugar right here. 2, 3, 4, and 5. OH, OH, OH. Okay. So this sugar is ribose. Specifically, this is D-ribose. Now, I have cyclized this. My hydroxide group is pointing upwards on my anomeric carbon, carbon number 1. It's an anomeric carbon because it is a new chiral center. It's pointed upwards. Sorry, just one moment. Sorry about that. So we've got D-ribose in linear form. And I didn't draw the hydrogens, but I'm going to go ahead and do so on my linear form because I do want to have a complete molecule there. Now, when I've cyclized this, this is a 5-membered ring. Now, if you remember, we have 5-membered and 6-membered rings. The 5-membered ring is a furan ring. So this is a furanose. So the complete name for this sugar right here is actually beta D ribofuranose. So beta D ribofuranose. Now, one thing that you've maybe noticed at this point in time when we talked about sugars is I presented the L isomer and kind of left it very quickly after that. And the reason for that is the L isomer of basically every single sugar is biologically irrelevant. So this is a point where y'all have the background information from organic, where you talk about positive and negative dextro and levo rotary molecules. Basically, how do they rotate, or how do they impact polarized light, or how are they impacted by polarized light? And here what we have is an instance where we say, here's your chemical knowledge, but we're going to proceed with the D isomer of this. So I always like to share this as trivial information, but D-glucose is your biologically metabolized isomer. L-glucose, by contrast, is not biologically relevant, and it's not going to be metabolized. However, it is sweet. So what that means is that let's just say L-glucose is something that you would taste the sweetness of it, but all of the enzymes in your body involved in sugar metabolism are going to basically ignore it. And so what that means is that if you had something sweetened by L-glucose as opposed to D-glucose, well, that substance, let's just say a soda, will be sweetened. So you'll get that satisfaction from it, but you won't have the caloric load. And so naturally, it's, I think, fair to say, well, why don't you just make sugars or sugary sodas sweetened by L-glucose? And the reason for that is 1 gram of L-glucose is is approximately $164. Now, if you think about a Dr. Pepper or something like that, a Dr. Pepper is, I think it's about 40 grams in 20 ounces. So your bottle of Dr. Pepper is 40 grams of glucose of sugar. It's actually fructose, but let's just say for this you'd be sweetened with glucose. Well, you have 40 grams of it. 1 gram is $164. That means that to sweeten that soda calculator, it would be approximately 40 times 164. You're talking about a $6,500 soda. So that's one reason why you don't have diet sodas that are sweetened with L-glucose. Um, at any rate, I wanted to bring that back just to reconcile D and L isomers of sugars and just to kind of make you fully aware of that. But with that in mind, this molecule right here, beta D ribofuranose, this is a very important molecule because this serves as your base sugar for nucleic acids, for RNA, ribonucleic acid, and modifying this gives you um, deoxyribonucleic acid. So that's what we're going to be talking about here. We're going to be discussing and delving into nucleotides and nucleic acids. So the basic nucleotide structure is shown over here on the right. It has 3 components: a phosphate, a base, and a sugar. The sugar that's depicted here is that ribose sugar that we previously talked about. Nucleotides, that unique name with a cap or with a T there, a nucleotide compared to a nucleoside. A nucleotide is composed of a nitrogenous base here, a sugar, and at least one phosphate. And that is mentioned as at least one phosphate because of the fact that, well, you can have a chain of phosphates. So here, or it could be 3 phosphates. So at least one phosphate. So the presence of those 3 components, phosphate, a nitrogenous base, and a sugar, make up a nucleotide. A nucleoside, by contrast, well, when we talk about a nucleoside, we're talking about something that consists of only a sugar and a base. So no phosphate is present. So what you would have is something like this. Now, of the different bases and base options, we have this umbrella term for our 2 classes of bases. We have pyrimidines and purines. A purine is a bicyclic ring structure. A total of 9 atoms make up the 2 rings. Our numbering starts at this nitrogen out here. And if you look at that and think, well, why is that the priority? Why is that the prioritized atom? Well, that all goes back to the biosynthesis of this molecule. Biosynthesis is something that you get into in your second semester biochemistry class, that metabolism class. In this class, we don't get into that. But here's our numbering scheme. We have that would be in counterclockwise fashion, we go 1 through 6. Then, in a clockwise fashion, we go 7 through 9 on the other atoms of the other ring, or of the remaining atoms on the other ring. A pyrimidine, we start at the 6 o'clock position where we have our nitrogen, and we go in a clockwise fashion 1, 2, 3, 4, 5, 6. So numbering is important because of the fact that there are nitrogenous bases that get modified, and they get modified by adding a specific, let's say, a methyl group to N7. And that modification can lead to a delay of converting DNA to RNA. So it can interrupt this, just as a hypothetical. And so it can delay the production of a protein. But numbering is important for that reason. Now, when we look at our purines, we have two specific purines, adenine and guanine. These are the names of those nitrogenous bases alone. Those are our two purines. Those have a shorthand notation of A and G. The pyrimidines, on the other hand, are the single ring structured molecules that are cytosine, thymine, and uracil. Again, if you look at the numbering, starting at that 6 o'clock position, and you go clockwise, I think this is the most effective way to learn these structures, which I'm expecting you to be able to look at the structure or look at a molecule and identify, is it adenine, guanine, cytosine, thymine? I think what's really productive is to recognize what makes these molecules different from one another. For instance, the 4 o'clock position of our pyrimidines at the, sorry, not the 4 o'clock position, the 12 o'clock position, but the 4 carbon on cytosine and thymine and uracil. That's where you have NH2 or O. If it's NH2, we're talking about cytosine. If it's O, then it's, or if it's a carbonyl oxygen, it's thymine or uracil. Okay. Then at the 5 carbon, what do you have? Do you have nothing or a methyl group? And I say that just because, or I say that as the two options, because you might look at this and think, oh, well, I've got to remember the double bond. I've got to remember this double bond. I've got to remember all these double bonds. Oh, man, I got to keep that straight in my mind. Oh, is this a nitrogen or is it hydrogen? And I would recommend not worrying about that, but instead recognizing, okay, 2 carbon, we've got double bonded oxygen, a carbonyl oxygen on every single one of these, cytosine, thymine, and uracil. At carbon number 4, we either have an NH2 or a carbonyl oxygen. At carbon 5, we have nothing or a methyl group. So if you remember those 3 things, you can go ahead and draw a simple structure that looks like this. And from that simple structure, say, all right, this is number 1, 2, 3, 4, 5, 6. Now, as soon as I put a methyl group at carbon number 5, which let me go ahead and interrupt myself, your exams are multiple choice, so you're not going to be expected to draw these. But I think for the purposes of preparing to recognize these structures, drawing is an effective study habit, study method, whatever. If you put a methyl group there, you know that this cannot be uracil or cytosine. And since it's not cytosine, you know that at carbon 4, you're not going to have an NH2, but instead you're going to have a carbonyl oxygen. And you know, since we're talking about a pyrimidine, at carbon number 2, you've got a carbonyl oxygen. Now, that to me conveys the important parts about this. In the event that you wanted to say, I need to get all of the details straight, I need to make sure that every atom has the exact number of bonds and hydrogens on it, then what I would recommend is just thinking back to Gen Chem 1 and the fundamentals that you learn from drawing Lewis structures in Gen Chem 1. Well, what do we know about nitrogen? Nope, please. Nitrogen likes to form this molecule right here. And whenever I say something like it likes to form something, I'm referring to it being uncharged. So it's a neutral molecule when it looks like that. So how many bonds does nitrogen like to have? It likes to have 3 bonds and 1 lone pair of electrons. So is there anything about the nitrogen at nitrogen at position number 1 or 3 that excludes that? Or is there any reason to think that it would, want to have a positive charge? When we look at these pyrimidines, and actually, heck, I'll go back and look at the purines. Do you see any complete charges? Do you see a positive charge on any one of these atoms? No, you don't. It's not, none of these molecules are going to have an overall charge. They might have slight partial charges, but they're not going to have an overall charge. So if you're looking at this and you're saying, okay, nitrogen at position number 1, I'm going to erase this just because it's a little bit cluttered. Well, this is either going to go down to a sugar or it's going to have a hydrogen. Now, that nitrogen at position number 1, does it have any overall charge? It doesn't. It's got 3 bonds and, if anything, a lone pair of electrons. So do I need to stress about drawing a double bond there? I don't. So I'm not going to. Now, position number 3, nitrogen here. Well, I have 2 bonds to it. And let's see, what should I do? Well, does it have a charge? It does not, because my bases, whenever I draw them, they have no charge. So I think the simplest thing to do would be to draw a hydrogen. And sure, I've drawn the bond between my nitrogen and my hydrogen, which is a little bit different from what's displayed there, but it's the same thing. It's a little bit different, but this is just a condensed visual, whereas what I've drawn is something depicting that bond. So nitrogen at position number 3, we got a single bond going from nitrogen to hydrogen. Okay. Now, if you were to go ahead and look at all of the other atoms, say carbon at position 2, does that have 4 bonds to it? It does. Carbon at position 4 has 4 bonds to it. Carbon at position number 5 does not have 4 bonds to it. Carbon at position number 6 does not have 4 bonds to it. So this is an instance where you can look at that and say, okay, well, it might be helpful or productive for you to say, okay, well, I know that this is a resonance stabilized molecule. And so if it's a resonance stabilized molecule, I'm going to have at least some delocalized electrons. Put that double bond right there. Carbon at position number 5 has 4 bonds to it. Carbon at position number 6 has 3 bonds to it. So I'm going to put a hydrogen there. And I'm good. That carbon at position number 6 has 4 bonds to it. Okay, so these are, I would argue, the staple information that is worth knowing to distinguish cytosine, thymine, and uracil. I recommend starting out with, I'm going to put double bonds between atoms, or at least one double bond between some of my atoms on my ring. But I'm going to look at carbon number 2, carbon number 4, carbon number 5, and say, all right, what am I putting there? Or or what is present there. If it's a methyl group, it's thymine. If it's nothing, then it's cytosine or uracil. So then I have to look at position number 4 and say, okay, what's present there? So look at, I hope that I said carbon number 5. I'm pretty sure I did. Carbon number 5, what's present? If it's a methyl group, it's absolutely thymine. If it's something else, well, if there's no methyl group there, then it's something else. So I think identifying the uniquenesses of these molecules is the most productive thing to do for understanding and knowing the structures. And that all boils down to and all goes back to Gen Chem 2, drawing structures, which you then developed in Organic 1. Okay, so now when we look at adenine and guanine, okay, so remember we had nitrogen at position number 1, and then we had a counterclockwise numbering scheme, and then we had a clockwise numbering scheme. Okay, so position number 1, then position number 2. Position number 2 is one point where we have a differentiation. So C2, what's present? Is it NH2 or not? Then at position number 6, C6, is it NH2 or carbonyl oxygen? If you can almost set this up as a flow chart, like C2, if it's an NH2, then you are looking at guanine. If you are looking at nothing, or yeah, then you are looking at adenine. Then you just have to remember, well, adenine has NH2 on C6. Where's the tablet? Okay, there we are. Guanine has at that same C6 double bonded oxygen. Okay, so this is all about the bases. And I'm really proud of myself for not making some lame Megan Trainor song. Maybe I'm not proud about that. I suppose I could be YouTube billionaire if I made some clever DNA inspired all about the base. Anyway, I'll move on. So you have basically a series of questions you can ask yourself when you're looking at a base. Is it one ring or is it two rings? If it's one ring, it's a pyrimidine. Now, what's my numbering scheme? Well, I start at the 6 o'clock position. That gives me my atom number 1, that nitrogen. Now I number them in a counterclockwise fashion, not counterclockwise, counterclockwise fashion. And I ask myself, what do I have at position number 4 and at position number 5? And as soon as I can answer that question, I have a way of triaging this information, processing which nitrogenous base I'm talking about. Now, did my computer come up? Yes, it did. Sorry, everyone. Then if I have a 2-ring structure, adenine and guanine are the 2 possible answers. My numbering starts in a larger ring where I have nitrogen, number 1. I go in a counterclockwise fashion, numbering them, and then a clockwise fashion. At carbon number 2 and carbon number 6 are where I have my different modifications. I think that's a productive way as opposed to simply memorizing these structures. It gives you insight into what structure you're actually talking about there. And that will be really helpful because whenever you go on to take metabolism, you'll get some insight into what the structures are and how they're built. So knowing these structures is very helpful for understanding how they're built. Now, with that in mind, um, these molecules are readily used as and derivatives of these molecules are readily used as anti-cancer therapeutics, because what they're going to do as anti-cancer therapeutics is they're going to interrupt metabolism and synthesis of DNA, synthesis of RNA, so interrupting the cell cycle, so getting uncontrolled cell replication basically to stop. That's the aim when you use any of these derivatives. Just one moment. So that captures the key parts of these structures in identifying these different molecules. Now, as we proceed, it's important to know that the keto forms of these bases are predominant forms. As you can see here, um, this is the keto versus the enol form. So this is what's known as a keto-enol tautomerization. So when we look at something like thymine, for instance, and let's see, this would be 1, 2, 3, 4. The predominant form of our molecule is this one right here. Now, what can happen is a tautomerization where this double bonded oxygen, this carbonyl oxygen, becomes an alcohol or an enol. And so we have that kind of transition that does take place. And the reason is that they can both occupy the same geometric space, but ultimately, if you were to like, I always like to imagine freezing time or freezing a molecule and looking at, well, what's the structure that is the most common? You're going to find that keto or lactam form of the molecule. So the ones that we looked at on the previous page are the ones that we're going to see. Now, second part of a nucleoside is the sugar. And I'm going to go from a nucleoside or a nucleotide, but I'm going to focus on kind of building a nucleotide. So when we have a nitrogenous base and a sugar, we've made up a nucleoside. In ribonucleotides, the pentose sugar is ribose. The same thing goes for in ribonucleosides. The sugar is ribose. In deoxyribonucleotides and deoxyribonucleosides, the pentose sugar is deoxyribose. The difference between those two sugars, as depicted here, or as, yeah, the difference between the two sugars is this right here versus this right here. So that is on our sugar, the sugar C2, carbon number 2, or the second position on our sugar. Now, one thing that you might notice as like, oh, well, is that important or not, is that prime symbol. So that apostrophe after 2. And that apostrophe is important. The reason is because if you think about the numbering that we just discussed with our purines and pyrimidines, our numbering was 1, 2, 3, 4, 5, 6. And then for our purines, it was 1 through 9. If I said position number 2 lacks an OH group in this deoxyribonucleotide, you have to have a concise way to say that and it to be known that we're talking about that position number 2 on the sugar. And the way that we differentiate is with that prime. So if you talk about position 2 in a nucleotide or a ribonucleotide or a deoxyribonucleotide, this is in reference to on the base. Whereas if you talk about 2 prime, that is on the sugar. So that prime numbering scheme is employed just to talk about the positions on the sugar. So if someone were to say position number 7 prime Well, looking at our sugars here, deoxyribonucleotides and ribonucleotides shown below, there isn't a seventh position. So that would be someone who, or that would be something that you'd need some sort of correction on. Now, with that in mind, that's the main differentiator between ribonucleotides and deoxyribonucleotides. Excuse me. Now, one thing that's worth noting is nucleotides and nucleosides. What are we talking about? We're talking about um, our most common forms, our deoxy and ribonucleotides. But what's our requirement for a ribonucleotide? Nitrogenous base, ribose sugar, and a phosphate. If you talked about a nucleotide, not a ribonucleotide, you need a base, a sugar, not necessarily ribose, and at least one phosphate. So those small details of like what's the requirement? A base, a sugar, and a phosphate. Are we explicitly saying that a nucleotide has to have ribose as the sugar? No, we're not. Could it be glucose based? Potentially. Is that going to be used in storing genetic material? No, it's not, because storage of genetic material is in the form of DNA or RNA. Now, on to nomenclature. Like I was saying, the prime numbers refer to the sugar, or the position on the sugar. The non-prime numbers refer to the position on the base, and the phosphate group may be attached to the sugar at either of two positions. Okay. Now, the phosphate group here that we looked at is in the 5' position. So that's one of the positions. The other position would be at the 3' position. So if you've taken a biology class, you've probably talked about directionality of reading DNA sequences as a 5' to 3' direction. And that is in reference to, well, the 5' on that carbon, and then the 3' or 5' on that sugar, and then the 3' on somewhere else within that polymer. So 5' and 3' are important. Now, with that in mind, let's look at abbreviations for a ribonucleoside 5-phosphate. Now, that's something that what I think you'll recognize is not redundant, but it's a weird sort of terminology. So it's important for you to know that a nucleoside consists of a base and a sugar. A nucleotide consists of a base, a sugar, and at least one phosphate. I personally have a tendency to use terminology that is nucleoside plus phosphate rather than saying a nucleotide name. And that's kind of what's depicted here. So what we've got is abbreviations for ribonucleoside 5-phosphates. Adenine, guanine, cytosine, uracil. These are the names of our bases. AMP, GMP, CMP, UMP are the shorthand abbreviations or the abbreviations for adenine monophosphate or adenosine monophosphate. ADP is adenine plus a sugar plus 2 phosphates. ATP is adenine plus a sugar plus 3 phosphates. Now, I'm going to relate these two items to one another. When you see ATP, what is implied there is you are talking about the sugar being ribose compared to deoxyribose. So ATP is a molecule that is not used in DNA synthesis. Meanwhile, or is not integrated as a part of DNA synthesis. DATP, when you have a deoxy sugar at that 2' position, DATP is in DNA synthesis. So That lowercase d out front is essential. Oh, goodness gracious. Oh, goodness gracious. Is essential for differentiating between, is this a deoxyribonucleoside or a ribonucleoside? So that does matter there. So DGMP, DGDP, sometimes it even feels a little bit silly, especially whenever you get down to DTTP, deoxy sugar or deoxyribose. Thymine is our base, and triphosphate. There are three phosphate groups there at that 5' position. Now, here what we've got is a comparison of ribo and deoxyribonucleotides. So the terminology or the specific name, let's take this one right here. Our base is adenine. Our sugar, look to the 2' position and see that, okay, the 2' position lacks an OH group, therefore it's deoxyribose. Then our phosphate is, there is a single phosphate on the 5' position. So this is deoxy mono, but what's our base? Our base is adenosine. The exact name for this is deoxyadenylate or deoxyadenosine 5' monophosphate. So one way to look at this is to say something like deoxyadenylate equals deoxyadenosine 5' monophosphate. So deoxyadenosine, that's the nucleoside name. 5' monophosphate is indicating that this nucleoside has a single phosphate on the 5' position. So that is the same as deoxyadenylate. Deoxyguanylate is the same as deoxyguanosine 5' monophosphate. Deoxythymidylate is the same as deoxythymidine 5' monophosphate. Deoxycytidylate is the same as deoxycytidine 5' monophosphate. I personally rarely use the full nucleotide name, but instead I use what is in the parentheses here. Just because I don't know why. Really, it's habit more than anything else. So it's important to recognize the things that are providing you with information and what that information is. So when you see adenosine, is adenosine the same thing as adenine? No, it's not. Adenine is the base. When you see that suffix, that OSINE, I want you to think, aha, that now I'm talking about a nucleoside. So adenosine is the nucleoside. So it's kind of implied almost, or it's almost a habit that, well, when we're talking about a nucleoside or a nucleotide, our sugar is either going to be ribose or deoxyribose. I think that's a very productive way to look at this information and to get the most out of it and kind of be efficient with it. So these are all names that I kind of, or that, not kind of, I expect you to be able to process, differentiating between a nucleoside and a nucleotide, knowing the details between and the differences between a deoxy sugar and a ribose sugar and a nucleoside that has ribose or a nucleoside that has deoxyribose. And really, it's not too challenging to identify if a sugar has deoxyribose. As soon as you see DEOXY, it's lacking a hydroxide group at the 2' position. The 2' position as opposed to the 2 position. So this figure here, or these figures here, I think are really good about kind of belaboring the point, showing you all of the examples. So if you want to go ahead and spend the time looking at these, then more power to you, go for it. But if you think, okay, well, I've got it, I know the subtle differences, I think I'm going to be all right, then go for it. And then proceed. Um, so then with that in mind, use this to reflect on this slide right here. So if I had DATP, the name that I'm going to use for DATP is deoxyadenosine 5' triphosphate. An alternative to this would be, well, if instead of the 5' triphosphate, we had the 3' triphosphate. This tells you where those phosphate groups are. Are they at the 5' position or the 3' position? And that's just something that you kind of have to learn by looking at these molecules or by looking at the nomenclature and being as explicit as possible. Okay, so this is basically just extracting that same piece of information that we looked at, but also goes through what are our identifiers. This 3-letter identifier for bases and nucleosides, it's pretty uncommon. Generally, A, G, C, U, and T are what are used to identify anything regarding sequencing of a DNA or an RNA molecule. If you know the base and you know that's a DNA molecule, here's what you need to know. The sequence is A, G, C, T, C, T, C, T, and so on. The nucleotide name, if you're looking at this and saying, oh, well, that's different. You told us deoxyadenylate, or sorry, adenylate versus adenylic acid. Well, this is the same as something like acetate versus acetic acid. Adenylate versus adenylic acid, they're effectively the same thing. The only difference is, well, is that group that is ionizable, protonated or deprotonated? Is there an ion or is it neutral? So adenylic acid is, excuse me, the phosphate groups would be protonated versus adenylate. The phosphate groups would be deprotonated, so they would be forming ions. So don't get it mixed up on that, and you should be all right there. And to be completely honest, because we are talking about physiological conditions at a neutral pH or round about a neutral pH, it's pretty uncommon that I will ask about adenylic or guanylic acid. Instead, I'm going to talk about adenylate or guanylate or deoxyguanylate, because that is the form of the molecule that is physiologically relevant. Okay, so here are a couple more different depictions. In this slide right here, we were talking about adenosine monophosphate. In this slide right here, we were talking about ribonucleoside 5' phosphates, deoxyribonucleoside 5' phosphates. Well, the 5' position is just one position that a phosphate can be added. A phosphate could also be added at the 3' position. Or if you're talking about ribose, it could be added at the 2' position. Or, and I wouldn't say less common, but another instance is a molecule like this, cyclic AMP. Adenine is our base. Our sugar is going to be ribose. We have a monophosphate. This C right here stands for cyclic. In this case, what we have is a phosphate group that is bound to both the 2' carbon and the 3' carbon. This makes adenosine 2,3 cyclic monophosphate, or 2'. Oh man, I made an error there. Adenosine 2', 3' cyclic monophosphate. This is an example of a molecule known as cyclic adenosine monophosphate. Cyclic adenosine monophosphate is a secondary messenger. It's a secondary messenger that is generated pretty readily for the purposes of stimulating breakdown of fats. Um, it's involved in tons of hormonal responses. And so cyclic AMP, this is an example of cyclic AMP. As you can imagine, well, as long as there's a hydroxide group, as depicted here, well, then you can make a cyclic form of a nucleotide. So there's also adenosine 3', 5' cyclic monophosphate. But these are just some of the other depictions of how that phosphate group can be linked to that sugar. There are a number of different molecules that are involved in metabolic processes, and I'm not going to go into this too much, but coenzyme A, nicotinamide adenine dinucleotide, or NAD+, and this is your oxidized form. Oxidized form of NAD or NAD compared to NADH. Then flavin adenine dinucleotide. These are all examples of molecules that have a ADP component. So they have adenine, 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 ribose, ribose, ribose. Then they have two phosphates that link them to kind of this big other chunk of a molecule. And these are all, or what I want you to take away from this is think about nucleosides and nucleotides as not simply being involved in DNA and carrying genetic material, but instead they're kind of a building block. Now, coenzyme A is in tons of metabolic processes of fatty acid breakdown, well, actually breakdown or synthesis. Um, so fatty acid breakdown and synthesis, they're also coenzyme A is also involved in uh, if you've taken a biology class, you've probably talked about the Krebs cycle or the TCA cycle, or and this is all the exact same thing. The Krebs cycle, the TCA or tricarboxylic acid cycle. Coenzyme A is involved in that. NAD plus and FAD are both electron carriers, and they basically, you could think about them as helping to generate ATP, that high energy molecule that is synthesized in the mitochondria, which the mitochondria is known as the, or is also known as the, and I paused there so that you could have the opportunity to say quietly to yourself, the powerhouse of the cell. You're absolutely right. Okay. So when we talk about a DNA and an RNA molecule, there are a couple of linkages that are really important. One is a phosphodiester linkage. That is the bond that is going to use a phosphate ion to connect two sugars to one another. So here's a phosphodiester linkage. Now, this is a phosphoester linkage. This is a phosphoester linkage. Hence, phosphodiester linkage. These phosphodiester linkages are what connect sugars to one another. Our primary structure is our way of looking at and reading a DNA or an RNA molecule. If you look at this molecule is aligned in the 5' to 3' direction, which is how we read a DNA sequence. So we have these phosphodiester linkages that connect a 3' carbon to a 5' carbon, a 3' carbon to a 5' carbon, and so on. Ribonucleic acid and deoxyribonucleic acid are the sugars that are present. The sequence of a DNA molecule and an RNA molecule are read in the 5' to 3' direction. This is similar to, just to extrapolate out, and actually let me take a little bit of a moment to pause and talk about, well, we have DNA, which is transcribed, or the process of converted. It is converted to RNA. RNA is then converted to a protein. The process of converting DNA to RNA is transcription, and RNA converted to a protein is known as translation. A protein sequence is read in what is known as the N to C directionality, which when we talk about protein structure, that will make a little bit more sense. But for the time being, I just want you to know DNA and RNA sequences are read in the 5' to 3' direction. And the kind of ironic and funny thing about DNA and RNA sequences is, despite everything that we've looked at, the important part about a DNA sequence is the sequence of the bases. So when you have a DNA molecule, all that you care about are the bases. And again, I'm proud of myself for not making a goofy Meghan Trainor joke. Not even really a joke, but a reference, which I suppose I just did. At any rate, there we go. So here what we have is an example of a molecule going in the 5' to 3' direction. What we care about is identifying the bases. And one of the things that I think is always important when you look at a molecule like this, and you know, a very low-hanging fruit question would be something like, is this a DNA or RNA molecule? And there is a correct answer for that. And that correct answer is known by looking at the 2' position. You can look at that 2' position and say, aha, that 2' position has a hydroxide group, therefore this is a ribose sugar, therefore it's an RNA molecule. Then, based on that, you can say, all right, well, what's my sequence? What's my sequence of base pairs starting from the 5' direction to the 3' direction? Well, it would be A, and then here you've got U or T. It's either uracil or thymine. Well, thymine is in DNA, uracil is only in RNA. And we know since our 2' position has a hydroxide group, this is an RNA molecule, and therefore this is going to be a uracil nitrogenous base. This is before you even dissect what the ring structure you're looking at is. So this molecule reads AUCG. That's the important takeaway from this. From the 5' to the 3' end, AUCG. Now, that can be depicted in a couple of different ways. One is a little bit redundant, and one is a little bit more concise. The redundant form would be this first one that's shown. PAUPUPCPG. Okay. There has to be a phosphodiester linkage between each of these bases. So these phosphates are really kind of redundant and unnecessary, which is why something like this is shown, PAUCG, because of the fact that, well, it is important to say if there is a 5' or a 3' phosphate. Now, if in this case we had a phosphate group down here, I'm drawing at the very bottom of the page, well, then you would read something like this, PAUCGP. We've got phosphates on the 5' and the 3' ends of this molecule. That gives you that extra little bit of information. But in this case, the way it's shown, you don't have that 3' phosphate group. All right, so here we have the double helix, or here we have a model of the double helix of a DNA molecule. Now, this was originally elucidated, or basically the most important information regarding the structure of DNA was produced by Rosalind Franklin. Then two other researchers took that work, and after Rosalind Franklin and published a paper that proposed the double helix as the structure for a DNA molecule. Rosalind Franklin sadly passed away before she could be recognized and awarded a Nobel Prize. However, the work that she did basically moved the field of biological research forward tremendously. And she's thankfully recognized and remembered quite often because of the X-ray crystallography work that had been done. Now, around the time that she did her crystallography work, Linus Pauling, who had already received a Nobel Prize previously in his career, he had proposed a triple helix of a DNA molecule, but ultimately it was Rosalind Franklin's work that led to the determination and the proposal of a double helix. Now, when we look at this double helix, this is referred to as a DNA secondary structure. The reason for that is it's higher order. There's more complexity to it. So it's not simply the sequence of the base pairs, but instead how those base pairs or how those bases pair up with one another. And what does this thing look like in 2 and 3 dimensions? So one of the things that is shown here is that there's this green or olive colored ribbon, and that olive colored ribbon corresponds to your sugar phosphate backbone, or your sugar and phosphate chains. Um, so that green ribbon corresponds to the sugar phosphate, uh, I guess, polymer, you could say. And that is on the backbone. That's on the exterior of this molecule. Sometimes, whenever people talk about a structure of a DNA molecule, they say that it has an overall net negative charge, or it has an overall negative charge. And the reason for that is those phosphate ions. Sugars have no charge whatsoever, but the phosphate provides it with a negative charge. The bases also, we talked about those having no charge. Now, if you look closely at the center of the molecule, you'll see C, G, G, C. So that is base pairing of the different bases, or pairing of the different bases. That's all in the interior of the molecule. These are hydrogen bonds that connect those different bases to one another. Those are on the inside. The backbones are considered to be anti-parallel. So one backbone reads 5' to 3', and the other is going to read in the opposite direction, 5' to 3'. So what ultimately this will do is this gives this molecule this kind of unique structure that provides a major and a minor groove. So those grooves within these molecules basically afford an opportunity for a protein or some other molecule to kind of come along and insert itself and just do a little inspection. What are the bases that I'm looking at that are near there? So when you think about, like, replication of a genome, well, those are proteins that are involved in kind of digging into those major and minor grooves and seeing the different bases that are present. Now we can look at a model of those different bases and how they pair up with one another. We can see that a 5 prime, here's one strand going in the 5 prime to 3 prime direction, and the opposite strand going in the 5 prime to 3 prime direction. Our bases are adenine and thymine. Those have a total of 2 hydrogen bonds to one another. Here is our depiction of adenine and thymine, just zoomed in. We've got hydrogen bond, hydrogen bond. Cytosine and guanine. We have hydrogen bond, hydrogen bond, hydrogen bond. So between A and T, you have 2 hydrogen bonds. G and C, or C and G, you have 3 hydrogen bonds. Now, when you look at those, what's stronger? Well, if all those hydrogen bonds are equal to one another in strength, well, the number is what's going to differentiate the strength. G and C, you have 3 hydrogen bonds, and T, you have 2 hydrogen bonds. And that's what's ultimately going to dictate the strength of that molecule. So if you have a molecule, a DNA molecule that's nothing but G and C, those are going to pair up with nothing but G's and C's. Compared to a molecule that's nothing but A's and T's, well, that will pair up with something that is nothing but A's and T's. So if you have a DNA strand that's 10 G long, then that is going to pair up with something. Let me do some erasing here. It's G, I'm going to erase the 10. I'm just going to say G, 5 prime to 3 prime. It's going to be C, 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 C. Okay. So this is 5 bases that pair up with one another. 5 times 3, 3 hydrogen bonds each. Oh, I zoomed in somehow. This is going to be 15 H bonds. Now, if you had the exact same thing at the top, I'm going to do some erasing. 1 strand, 5 prime, 3 prime. A, 3 prime. T, 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 T. Well, that's going to be a total of 2 hydrogen bonds between each of the bases. We've got 10 hydrogen bonds here. So the length of these two double-stranded pieces of DNA are identical, but the number of hydrogen bonds between them is going to differ. So as we previously said, the keto form of the bases is going to dominate, which is why we're going to note that hydrogen bonding. Both sets of base pairs are going to occupy the same geometric state. And we have this base pairing of A pairs with P and G pairs with C. This is known as the Chargaff rule. And I cannot remember the first name of the person, but I guarantee you their last name was Chargaff, because you make a profound discovery, you slap your name on it. At any rate, that was our sort of here's our secondary structure. Here's a kind of flattened image of that secondary structure. RNA structures, by contrast, are much more complicated. I know if you've taken any sort of biology class, one of the things that will be said is DNA is double-stranded, RNA is single-stranded. And so you operate under this idea that this right here, these are our 2 strands, and then that's the hydrogen bonds between our base pairs. That's DNA. And we say, yay, DNA, smile. Then you look at an RNA molecule and you say, oh, well, it's a single strand. You say RNA. Not smile or frowny, but instead, what do I say about RNA? It's single-stranded. But it's hydrogen bonds that are going to pair up with one another. In an RNA molecule, single-stranded RNA molecule like this, when it is long enough, what it's going to do is actually form much more complicated structures. It's going to form, here's our 5' end to this to the 3' end. This is one single strand. It's going to form a number of different helices. It's going to form bulges and hairpin loops and internal loops where you're going to have regions that pair up perfectly and then regions that don't pair up perfectly. For instance, if you look at this, we've got a bunch of G's and C's. That's a lot of hydrogen bonds that are going to kind of cluster together. And then right next to that, you got 1, 2, 3, 4, 5. We've got 5 base pairs or 5 bases that are opposite 1, 2, 3, 3 base pairs. How are 5 bases going to pair up with 3 bases? Well, they're simply not. So that's where you're going to get these loop or odd structures in an RNA molecule. So a single stranded RNA molecule makes really crazy cool structures. And those crazy cool structures have been, just a second, I have to sneeze. Um, those crazy cool structures are basically the foundation for RNA interference and CRISPR technology, because what you can do is basically make a or design a molecule that eliminates a specific or eliminates an RNA molecule. So you can design something that targets another molecule for dicing and slicing up. And that's all through single stranded RNA. Now, a fair question is, why do we have both DNA and RNA? One way to look at it is, and the simplistic way that I was always kind of taught and thought was, well, a single stranded RNA molecule is unstable. A double stranded DNA molecule is considerably more stable. An RNA molecule is going to be degraded in a matter of hours on a benchtop. There's RNases everywhere, and RNA can hydrogen bond with everything because it's got those base pairs open and accessible to the environment. So they're going to hydrogen bond to things and they're going to break up. DNA, on the other hand, is super stable. But the differences between them, the sugars, the bases. Why does DNA contain thymine? And why is 2-deoxy and RNA is not? Well, that OH group at the 2' and 3' positions in RNA make it more susceptible for hydrolysis. So this is something that it simply evolved to form a DNA molecule. So this evolution led to the formation of a DNA molecule that would harbor that genetic material that RNA would not. When DNA lacks that 2' OH group, well, then it's more stable. Now, with that in mind, I'm going to go ahead and use that as our stopping point for part 1 of nucleic acids, because where we're going to go from here is into DNA technologies.